A single tablespoon of vinegar before a meal improved insulin sensitivity by 34% in pre-diabetic patients. That was published in Diabetes Care in 2004. Roman soldiers drank it every day on campaign. Hippocrates prescribed it in 420 BC. It was still listed as medicine in the British Pharmacopoeia in 1898. When researchers put commercial vinegar tablets head-to-head against liquid vinegar, the tablets failed to produce the same metabolic results. The most viral weight loss study ever published about it was retracted after its data was found to be statistically implausible. And almost nobody noticed when a 5,000-year medicine got quietly reclassified as a condiment. The 5,000-year record. The story starts in Babylonia, around 5000 BC. Someone left grape juice unattended. It fermented into wine. Then it fermented again into something sharper and more stable. The Babylonians did not throw it out. They used it to preserve food, the first recorded use of vinegar in human history. 2,000 years later, in ancient Egypt, vinegar residues were turning up inside sealed burial urns. The Egyptians were using it to pickle vegetables and fruits, not as flavoring, but as preservation. The same logic the Babylonians had followed. Vinegar stops spoilage, extends the food supply, keeps people alive. Then comes Hippocrates in 420 BC in Greece. He prescribed vinegar for cleaning wounds. He also prescribed oxymel, a mixture of honey and vinegar, for persistent coughs. That formulation worked well enough that it stayed in continuous medical use for over 2 millennia. By 1872, it was in the German Pharmacopoeia. By 1898, it was in the British Pharmacopoeia and the French Codex. A preparation that began in ancient Greece was still official medicine in Victorian-era Europe. That is a 2,300-year clinical record. The Roman military ran on it. Posca, vinegar diluted with water, sometimes with salt and herbs, was the standard daily drink ration of Roman soldiers for centuries. The writer Vegetius documented it directly. Soldiers should have corn, wine, vinegar, and salt at all times. Emperor Hadrian drank it alongside his troops to show solidarity. Cato the Elder kept drinking it after his military service ended. In 360 AD, a formal imperial decree ordered lower ranks to drink posca and wine on alternating days. This wasn't tradition, it was logistics. Acetic acid kills bacteria in uncertain water. On long marches, drinking from rivers and streams, Roman soldiers had a biological advantage built into their daily ration. And to reclaim more of these lost truths, subscribe to Grassling. Because while the Romans used this for survival, 10th century physicians found an even more specialized use. In 10th century China, a physician named Sung Si, the founder of forensic medicine, prescribed hand washing with sulfur and vinegar before performing autopsies. That is a documented antiseptic protocol, 800 years before Louis Pasteur explained what bacteria were. In France, the city of Orléans became the capital of Western vinegar making. The vinaigriers, the vinegar makers, had formal guilds, regulated production standards, and their own coats of arms. The Orléans process, with its slow barrel fermentation and carefully maintained living cultures, was the gold standard of the craft. Vinegar wasn't a byproduct or an afterthought. It was a respected trade with centuries of accumulated knowledge behind it. For centuries, the living culture inside those barrels was the protected knowledge of the guilds. Then, in 1823, a single patent filing in the Kingdom of Baden began to dismantle a record that stretched back to the dawn of civilization. A German industrialist named Carl Sebastian Schusenbach filed something new. Not a ban, not a seizure, just a faster, cheaper method that made everything before it economically indefensible. How a medicine becomes a condiment. Schusenbach's invention was called the packed generator. It circulated alcohol over beechwood shavings inside a tall wooden column. The shavings gave Acetobacter acetibacteria more surface area to work on, and fermentation that once took several months now took 1 to 2 weeks. That first compression made something newly viable, spirit vinegar, made not from fruit or grain or wine, but from pure industrial alcohol, acetic acid and water. Nothing else. The economics were simple. Factories could produce in weeks what traditional makers spent months building. The price collapsed. German industrial vinegar flooded European markets. The guilds in Orleans could not match it on volume or cost. They had centuries of accumulated craft and not a single competitive advantage left. By 1914, only 17 traditional vinaigreries survived in Orleans. There had been hundreds. The guild, with its own coat of arms, with its regulated standards and its carefully maintained living cultures, was economically finished. Not banned, not seized, just priced out of existence. Even Dessau Fils, the largest surviving maker, eventually abandoned traditional methods for industrial ones. The knowledge did not burn. It became unprofitable, which is often the same thing. Then came the second wave. In the late 1940s, two researchers at the Vienna University of Technology, Otto Hromadka and Heinrich Ebner, developed what became known as the submerged fermentation process. They borrowed the principle from wartime penicillin production: force air through a liquid medium in a sealed tank, accelerate bacterial activity, compress the timeline. It worked. Vinegar production dropped from weeks to between 24 and 48 hours. The packed generator had already cut months down to weeks. Submerged fermentation cut weeks down to a single day. Supermarkets demanded a clear, dead liquid that would never change on the shelf. To get it, factories had to take the most medicinal part of vinegar and reclassify it as industrial waste. The mother, the cellulose biofilm that traditional makers had passed down for generations, the living culture at the heart of the craft, was filtered out. For clarity, for shelf stability. A living thing, maintained in continuous use since before the Roman Empire, was simply removed from the equation. What came out the other end was technically still vinegar. It met the legal definition. It contained acetic acid. It was sour. But distilled white vinegar, what most households now keep under the sink, is acetic acid diluted in water and nothing more. No mother, no polyphenols, no enzymes, no organic acids. 15 or more beneficial compounds were filtered, pasteurized, and processed out of existence in the name of a clear, stable, cheap product. No one ordered this. There was no meeting where someone decided to strip vinegar of its medicine. The suppression was structural. It followed money and regulatory logic, not malice. Here is how that logic worked. When pharmaceutical regulation developed through the 20th century, it sorted everything into categories. Medicines required clinical trials, regulatory approval, and patented formulations. Vinegar was classified as food. It could not be patented. Anyone could make it. It could not generate pharmaceutical returns. So no drug company would fund the trials needed to validate what Hippocrates already knew. The research that does exist came from universities, not industry. There was no financial incentive to prove vinegar worked, so the proof was slow to accumulate. The funding dead zone did the rest. That same logic eventually reached the last surviving holdouts, the small producers still making living culture vinegar the traditional way. When industrial consolidation could not price them out, private equity moved in. The modern chapter of this story has a name: Bragg Live Food Products. Founded in 1912, Bragg became the most recognized raw, unfiltered apple cider vinegar brand in the world. Patricia Bragg ran it for 65 years as a family mission. Then, in 2019, a group of celebrity investors and a private equity firm saw something in that bottle that the original founders never did. Swanda Pace Capital acquired the company, with co-investors that included singer Katy Perry, actor Orlando Bloom, and pressed juicery founder Hayden Slater. Patricia Bragg, then 90, stepped down. Consumer backlash followed quickly. Customers reported the product looked lighter, different. Videos went viral claiming the recipe had changed. Bragg denied it. By June 2024, the company was exploring a sale at a valuation of over $500 million. What actually changed inside that bottle is disputed. What is not disputed is the pattern. A living culture product enters the private equity pipeline. and the people who built it leave. Nobody planned for vinegar to become a condiment. Industrial logic does not need a plan. It just needs a price advantage, a shelf standard, and enough time. Over about 150 years, a 5,000-year medicine became a shelf filler beside the ketchup. Part 3. What the research actually says. The evidence was never hidden. It was sitting in peer-reviewed journals, waiting for someone to look. Start with what vinegar actually is at the biological level. Fermentation happens in 2 stages. First, yeast converts sugars into alcohol. Then, Acetobacter bacteria convert that alcohol into acetic acid. That acetic acid is the compound behind nearly everything documented about vinegar's effect on the human body. The concentration in household vinegar runs from 4% to 8%. That's enough. The mother of vinegar is a cellulose biofilm, a thin, gelatinous film produced by Acetobacter during slow, traditional fermentation. It forms on the surface of the liquid as the culture matures. It contains live Acetobacter bacteria, enzymes, and a range of polyphenols, the same class of antioxidant compounds found in green tea and dark chocolate. Industrial filtration removes all of it. What you get in a clear bottle is the acetic acid, stripped of everything else that slow fermentation built. In 2004, Carol Johnston and her team at Arizona State University published a study in diabetes care. The finding was this: a single tablespoon of vinegar consumed before a high-carbohydrate meal improved insulin sensitivity by 34% in pre-diabetic subjects and by 19% in people with type 2 diabetes. 3 groups were tested: healthy adults, pre-diabetics, and type 2 diabetics. Every group improved. The pre-diabetic result was the sharpest. 1 tablespoon. Before a meal. Millions of people now take vinegar in pill form to avoid the sour taste. When researchers finally put those pills into a clinical trial against the liquid, they found a mathematical gap the supplement industry had never mentioned. Fies and Johnston, publishing in the Journal of Dietary Supplements in 2020, compared liquid vinegar directly against commercially available vinegar tablets. Liquid vinegar cut the 60-minute post-meal blood sugar spike by 31%. That's the difference between a post-lunch energy crash and a steady afternoon. The pills produced no equivalent effect. The reason is straightforward. Vinegar tablets on the market contain as little as 45 milligrams of acetic acid per dose. The effective amount delivered by the liquid in the Johnston trials is 1,250 milligrams. That's a 27-fold difference. The pills aren't a lower dose. They're a different thing entirely. In 2017, Shishabor and colleagues published a meta-analysis in Diabetes Research and Clinical Practice. They reviewed 16 separate trials. Across all of them, vinegar significantly reduced fasting blood glucose in the groups that consumed it. The authors concluded it could function as an effective addition to therapy for improving blood sugar control. Not a replacement. An addition. That framing matters. It is honest about what the evidence actually shows. A 2023 study out of Iran, published in Frontiers in Clinical Diabetes and Healthcare, ran a controlled trial in diabetic patients. Apple cider vinegar consumption significantly reduced fasting blood glucose, HbA1c, total cholesterol, LDL cholesterol, and the LDL to HDL ratio. Multiple metabolic markers, all moving in the right direction from the same intervention. The antimicrobial data goes back to basics. Wine vinegar kills Vibrio cholerae, cholera bacteria, within 1 to 2 minutes of contact. Acetic acid disrupts bacterial cell membranes directly. This is the mechanism. It is why Roman soldiers, drinking from rivers across North Africa and the Middle East, had a measure of biological protection built into their daily ration. On weight management, the picture is real, but modest. Kondo and colleagues published a 12-week randomized controlled trial in 2009, funded by the Japanese vinegar producer Mizkan. Obese adults who consumed 15 or 30 milliliters of vinegar daily saw statistically significant reductions in body weight, visceral fat, waist circumference, and serum triglycerides compared to a placebo group. The effects reversed 4 weeks after they stopped. Small but measurable. Repeatable under controlled conditions. Contrast that with the 2024 study that generated millions of views. Dramatic weight loss claims in Lebanese adolescents, published in BMJ Nutrition, Prevention and Health. That study was retracted in September 2025. The retraction cited implausible statistical values, unreliable raw data, and no prospective trial registration. The supplement industry is still marketing based on it. The real data, the Kondo data, and the Johnston data was always more useful. It just was not as loud. Part 4. What you can do with this. Here's what you can actually do with this, starting today. The protocol the Johnston trials used is simple. 1 to 2 tablespoons of liquid vinegar, 15 to 30 milliliters, diluted in a full glass of water. Drink it before or with a high carbohydrate meal. Because every vinegar contains acetic acid, any vinegar works. The pills do not, like you learned earlier. The reason liquid vinegar affects blood sugar comes down to a single enzyme called AMPK. Think of it as a master switch for how your cells handle fuel. It sits in your muscle and liver tissue, and acetic acid flips it on. When AMPK activates, it mimics what exercise does at the cellular level. Muscle cells pull more glucose out of the bloodstream, and the liver releases less of it. That's the mechanism and why the number moves. Now here is the part the supplement industry does not want you to think about. You can make the real thing at home from kitchen scraps for essentially nothing. You need apple cores and peels, the parts you would otherwise compost, enough to fill a wide-mouthed glass jar about 3/4 full. Add 1 tablespoon of sugar for every cup of filtered water you pour over the scraps. The sugar feeds the initial yeast fermentation. Cover the jar with a breathable cloth, a coffee filter or tea towel works, and fix it with a rubber band. Do not use cheesecloth. Fruit flies will find it. For the first 3 weeks, stir the mixture once a day. This keeps mold from forming on the surface. Keep the jar somewhere dark and warm, between 65 and 80 degrees Fahrenheit. You are running the first fermentation here, yeast converting the sugars to alcohol. At week 3, strain out the apple solids and compost them. What is left is a rough alcoholic liquid. Now the second fermentation begins. Cover the jar again with the breathable cloth, set it back in its warm corner, and leave it alone. Sometime between week 4 and week 12, a white film will appear on the surface. That is the mother, Acetobacter, building its cellulose biofilm, converting the alcohol to acetic acid. Do not disturb it. It is working. Start tasting at the 2-month mark. When it is sharp enough for your preference, bottle it. Raw apple cider vinegar has an indefinite shelf life because of its own acidity. No refrigeration needed. Store it at room temperature out of direct sunlight. The acidity keeps it stable, but light and heat break down the polyphenols that slow fermentation. The most important step comes after bottling. Save the mother. Pull it out gently and keep it in a small jar with a little finished vinegar. When you are ready to start the next batch, add it. The mother inoculates the new liquid and cuts weeks off the timeline. One batch seats the next. That is how this culture survived for millennia before industrialization made it inconvenient. The yield is roughly 1:1. A half-gallon jar of apple scraps and water produces about a half-gallon of finished vinegar. If you are using scraps from cooking, the cost is effectively 0. A bottle of raw apple cider vinegar with the mother at a grocery store runs $5 to $8 per quart. Your version costs the same as the water. One caution worth stating plainly: always dilute before drinking. Undiluted vinegar damages tooth enamel and can irritate the esophagus over time. And if you are managing diabetes with medication, this is a complement to that treatment, not a replacement for it. The research is real. so are its limits. You don't have to be a vinegar customer. You can be a fermentation practitioner with a 5,000-year lineage behind you. From Roman field rations to Hippocrates, to the mother that could be floating in your jar, doing exactly what it has always done. That culture is alive. It reproduces. You can give it to someone else, and the lineage continues. That's how real knowledge has always traveled, not through supply chains, but through hands. Subscribe to Grassling. Share this with someone who is ready to stop outsourcing their health to systems that were never built to make them truly well.